Good evening. Che. Yes, I am clearly audible. Hello, good evening, my dear children. So someone stole my pen, by the way. Just give me a second. Oh, it was me only. Your Jasmine Ma'am is... Okay. Akash is saying translate this. Dear, when I click on it, it comes hi. Okay. So you said hi. A big hi to Akash and a big hi to everyone who are waiting for this particular class and everyone who probably are going to accidentally come into this particular class as well. My dear children, I hope every one of you who is watching this video is doing absolutely good, and I really hope you have watched the part 1 of this particular video. That is equally important. And by the way, if any one of you feels that this chapter is super difficult, which is why probably I'm gonna skip this off. Okay, if at all you have a plan like that, do not do that, because this chapter gives you 13 marks in your PUC boards. And not just that, in your KCET it gives you highest number of questions, that is almost 5 to 6 number of questions are there coming from this chapter. Hello Rekha, how are you? I hope you're doing good. And Akash, I translated Macha, I got the thing. It's hi, you said hi, right? So hi to you as well. Okay, so we'll just wait for a second so that everyone else also can probably slowly come and join. And my dear children, I'll request you can also share it to your friends, and probably they can also come and join the class. Okay? Perfect. So now let's go. Slowly let them come. So the first thing that we're going to deal with today is going to be the dihybrid cross experiment, a very important topic. Children get confused about it way too much, which is why I thought we should be dealing it with first. So in the previous session we studied about the monohybrid cross. We saw how the when we cross tall plant and dwarf plants, how do you get all of them to be tall, and when you cross them again you get tall and dwarf both. Mendel's 2 law of inheritance, that is law of dominance, law of segregation, we studied about that. And what all did we study? We studied about the exceptions of laws of inheritance and all of it. So Rekha and Akash, I hope we are super ready for this particular topic, that is dihybrid cross experiment. Ready? If ready, give me a thumbs up. If ready with this particular thing, can you quickly give me a thumbs up so that I understand that yes ma'am, we are ready. Yes, perfect. Chalo. So dihybrid, what do you mean by that? By the way, you get 5 marker question from this particular concept in your second PUC. They directly ask you, represent schematically the result of 2 gene interaction by taking seed shape and seed color. So it's a 5 marker question that I'm going to deal with right now by explaining this. Listen to me very carefully. Dihybrid basically means, until now we spoke about only one character, that is what? Height. Tall and dwarf comes as a subtype of height, right? But now we are going to talk about 2 characters at once, and we're going to see how these 2 characters are going to pass from one organism to the other organism. 2 characters at once, which is why I call it di. Di means 2. Okay, why do I call it hybrid? Because while you're going to do all of this, you're going to see new types of plants being formed. Okay, that is why you call this as dihybrid cross. And in dihybrid cross, the most famous thing that I have done over here is that I have taken 2 conditions. Okay, one is going to be the shape of the seed. One is going to be the shape of the seed. It can be either round, okay, or probably it can be wrinkled. If it's round, it looks very cute, okay, it's just like this. If it's wrinkled, it looks like this. Okay, that is what you mean. Now, the next character that I took was basically the color. I'm talking about seed shape and seed color, okay? In color, we had 2. Anyone can guess the 2 of them? Anyone? If at all you remember, in previous session, we studied about gravity. That is basically not gravity in physics, but a short form of all the characters that were there, no? Of all the traits. If at all you remember, in color, seed color, you have 2 different type of colors. In the case of garden pea, what are those 2 different colors? Any idea? Ma'am, I know the answer, but I will not tell. Ma'am, I do not know the answer, which is why I will not tell. Either of the category you guys are in, you can simply guess, not an issue. Okay, now I believe that Akash and Rekha, both of them are very shy to give the answer. So probably I'll only end up, because if I become shy, then nothing works, right? So over here, the color can be either yellow, 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 dirty fellow, or it can be green. Or it can be green. Okay, in yellow and green, which one is going to be dominant? This is dominant one, and this is going to be recessive one. Okay, similarly in round and wrinkled, this is going to be the dominant one, and this is going to be the recessive one. Wrinkled is going to be the recessive one. Okay, okay. Now we are going to make the parents. In the parents, I am saying that the first parent that I have is round and yellow. I am saying it is what? Why is not getting clicked? Just a second. It is round and yellow, and it is pure breed round and yellow. What do I mean by pure breed round and yellow? That means both the alleles are going to be same. For round, what do you have? You have capital R and capital R. This is what I mean by pure breed. Both the alleles are going to be same. For round, we have that. For yellow, how will you denote? Y and capital Y. Both of them are same, right? Yes. Now we are going to cross it with a plant that is having wrinkled and green seeds. Wrinkled, if round is denoted by capital R, wrinkled is going to be denoted by what, Baba? Small r. And here also it's pure breed, so both of them have to be same, small r, small r. What about green? Green is also going to be, here it is capital Y, then here it is going to be small y, small y. These are the parents that I have. What are they? They are pure breed. They are homozygous. That is what I mean by pure breed. Okay, now we want to form the gametes. And if you understand this particular part, you have already cracked the whole concept. Listen to me carefully. Now when I say that we want to form a gamete, always imagine this to be something like this. Suppose you have a chromosome right over here, a pair, and over here another type of chromosome. On one chromosome you have capital R allele. Okay, on one chromosome you have capital R allele. Here also you have capital R because you have two capital R, capital R. This is what you mean. You have two chromosomes where you have capital R, capital R. Now what about this? This is going to be having capital Y, capital Y. Cool. This is how it looks for this particular parent. Now what about over here? Here also we will make one chromosome. Here also we are going to make one chromosome. The gene here smaller, here also smaller because that is how it was there. Similarly over here also we are going to make something like this, and we are going to make small y and small y. Now we can make the gametes. In the gamete, Baba, we have studied in law of segregation. From one pair, only one guy goes to the gamete. Suppose from here anyone goes. It will always be capital R. Whatever you do, it is going to be capital R because it does not have anything else to give, right? Similarly over here, whatever you do, it will be just capital Y only. It will be just capital Y only. So when you form the gamete, you can just imagine a chromosome containing capital R, a chromosome containing capital Y is going to go from here. Okay, whatever gametes, however number of gametes you want to make. Similarly, if you consider this particular scenario, what is going to happen? From this pair you get small r, from here you get small y. That means that a chromosome carrying small r and a chromosome carrying small y. This is how it looks. This is how all the gametes are going to look. Now we are going to do fusion. Okay, the gametes which are there, we are going to fuse them up, and we are going to see what do we get in the first generation. Anyone who is watching this video, can you guess it? What do we get in the first generation? When you have when you have two gametes which you are fusing like this, what do you get in the first generation? I am gonna go ahead, but if at all you wanna try, you probably can try right now. Listen, I am full energy. Children, if at all you are not participating, then this is going to be a boring game. You have to participate, and trust me, I will not remember you giving me wrong answers. Promise. Wanna try, or shall I only go ahead? No? No one? Akash? Rekha? Anyone who is hidden up there and not showing up over here in the chat session? Don't want to try? Then I'll go. Listen to me carefully. Capital R. The first generation, what do I get? Capital R from here, small r from here, capital Y from here, small y from here. Now you have to use your imagination, and it's very, very simple. Listen, you had got this chromosome containing capital R, right? You're going to get chromosome from this gamete also. This time it is containing what? Small r, right? From here you get a chromosome carrying capital Y. From here you're going to contain a chromosome carrying what? Small y. So that is how we get something like this, that is capital R, small r, capital Y, small y. Now, what is the phenotype? How do they appear? This is the genetic makeup. This is what? This is the genotype of F1 generation. This is the genotype of F1 generation. I'm asking you, what is the phenotype like? How does it appear? This thing, how does it appear to you? Does it appear pink and green to you? Does it appear round and yellow to you? Does it appear wrinkled and green to you? How does it appear? That's the question. Come on, put it in the chat session. Come on, Macha, I'm waiting for that one child to go ahead to lead and say that, ma'am, this is how it looks. No? Don't want to try? Still shy? Then probably I'll only give the answer. Okay, let me go ahead then. Okay, it is going to be, listen, capital R is dominant over small r, and capital R was denoting what? Round. So it is going to be round for sure. Okay, what about this? Capital Y is going to be dominant over the small y out there. Capital Y was for what? Yellow, which is why it is going to be yellow. All of them are going to be round and yellow, but the genotype is going to be like this. This is genotype is known as what? Heterozygous. They are heterozygous. This was homozygous, round and yellow. Both the alleles were same. This was homozygous. This is heterozygous, round and yellow. Okay, this is going to be heterozygous. Why? Because the alleles over here are not same. Cool. Now Mendel, while doing his experiment, he found that when I crossed round and yellow plant and basically wrinkled and green plant, I found all of them to be round and yellow. What about the wrinkled and the green? The same thing I noticed when I was trying, suppose I am Mendel, okay? I was crossing tall and dwarf, I saw all of them were tall in the F1 generation. Then I did one thing, I did selfing. I crossed among themselves, then I found my character. Similarly, I am going to do that over here also. The same type of progenies are going to develop. I am going to cross them. I am going to cross them. And let's see what is going to happen. And this time you have to participate. Otherwise, it is going to be super boring. Okay. So I am taking the parents. The parents looks like this. Capital R, small r. Capital Y, small y. Similarly, capital R, small r. Capital Y, small y. Now we are going to make the gametes, Baba. Let's see how the gametes are going to look. Okay. Before we make the gametes, we have to use our imagination. One chromosome capital R, another chromosome small r. One chromosome capital Y, another chromosome small y. Similarly over here also, this parent also same thing will be there. Okay, one chromosome capital R, another chromosome small r. One chromosome capital Y, another chromosome small y. Okay, now when we form the gametes, what is going to happen? From this pair, one guy can come. That is the rule. In the gametes, only half of the chromosome can go, right? So from this pair, only one can come. So suppose the first type of combination is capital R. From here also only one can come. Suppose capital Y came. That's all. Is that the only gamete that we form? No, there can be multiple combination. This is one type. Next type. Suppose from here capital R came and from here small y came this time. This is the second type of gamete that we are forming. Okay, next type. From here small r came, from here capital Y came. Third type of gamete, right? Now one more is there. From here small r came and from here small y came. 4 different type of gametes we have formed. Does this parent have something else or a same parent only? No. Same type of gametes are going to be formed. Same type of gametes are going to be formed, right? So here also we got same gametes. There also we are going to get the same gametes. Now we cannot keep crossing them like this, like this. It becomes a lot of big task. Yes, sir, man. Okay, so what are we going to do? We are going to arrange them over here. We are going to arrange 4 gametes over here. We're going to arrange 4 gametes over there, and we are going to cross them. And we're going to see what is going to happen. Until this particular point, is everything clear? If everything is clear, can you give me a quick thumbs up? If everything is super clear, can you give me a quick thumbs up? Not clear, can you give me thumbs down? Oh my God. Okay, Akash gave me thumbs up. Thank you so much. Cool. So we had made 4 gametes here, 4 gametes here, and we're going to fuse them out. Ma'am, can I take a drop for this year for KCET? Nat Bhaikar Dali. Dear, if at all you feel that is very much necessary and probably you are not getting a very good college and you do not have any options left behind, I'll request you to still look out for other courses. If that also does not work, you are totally inclined for something else, yes, you can go ahead and take a drop. Okay, but you have to be ready for it. Taking a drop is not going to be an easy process. Okay, you have to invest time, you have to be consistent. So that's why I'm telling you. Probably give some days, okay, and try to search whatever you can do, whatever you feel to do at this particular point of time. If it still fails the same, then you can take a drop. Cool? Yes. Now I'm coming back. So when I cross this and this, what do I get? Capital R, capital R, capital Y, I got round and yellow. When I cross this and this, what do I get? Capital R, capital Y, small y, still round and yellow only because Y is dominant over that. Okay, when I cross this and this, capital R, small r, capital Y. Okay, this is going to be still round and yellow Y because this is dominant over the other one, right? And finally, when I cross this and this, what do I get? Capital R, small r, capital Y, small y, still round and yellow. So round and yellow, you have different types of round and yellow being formed over here. But you get it. How many progenies are there? Almost 16. 4 into 4 basically. You are going to see 16 combinations. Okay, you are going to see 16 combinations out there. And only one of them is going to be wrinkled and green. So now I'm going to decode that. We are going to talk about the phenotypic ratio, which is super important in terms of the dihybrid cross. Okay, so the phenotypic ratio goes like this. All the 16 progenies that we have got, physically if at all we see, they look different. They are going to look different. There is a ratio that is 9:3:3:1. 9 of them are going to look round and yellow. That means the dominant parental trait. We started off with round and yellow. So this one, 9 of them are going to look round and yellow only. One of them is going to show the recessive parental trait. What was it? It was wrinkled and green. So let's make that wrinkled and green. Okay, 3 of them which are there and 3 of them which are there, they are not going to be like the parents. Okay, they say that I cannot be like the parents. I have to be very different. So let's see that. What is going to happen? So they were round and yellow, wrinkled and green. So if you do not want to be like your parents, round was always with yellow. This time it is going to be with green. Similarly, wrinkled which was there, it was always with green. This time it is going to be with yellow. So what do you see? You see that 9 plus 1, 10 of them are going to be like your parents. So I'll write parental in nature. Okay. Whereas 3 plus 3, 6 of them, my dear children, they are not going to be like your parents, going to be like the parents out there. So it is going to be non-parental. You also sometimes call this as recombinants. What do you mean by recombinants? They are new combinations. Recombinants means new combination. Revision means again you are revising something. That is what you mean, right? So they are going to be recombinants. So in total, if I have 16 progenies in together, 10 of them are going to be like the parents, 6 of them are going to be non-parental. Done. And if at all you ever forget the ratio 9:3:3:1, just look. 9 of them are going to be what? Round and yellow, dominant parent trait. 1 of them going to be recessive parental trait. And in between, you form the new combination, round with the green and wrinkled with yellow. Got it? That's all. Is this particular part also clear to you? Is it possible to score 100,000 rank, ma'am? Definitely. Is that even a question? You can go ahead. And if you give your best, there is under 1,000, you can probably get rank 1 also. Okay. So children, clear? This particular portion is also clear. If clear, can you give me thumbs up? If this portion is super clear to all of you, can you give me thumbs up? Probably I can go ahead and make you solve some questions from KCET. Done? Okay. So I'll go ahead and probably while we solve questions, move on, ma'am. Thank you so much. Okay. So did Gregor Mendel find something unique while he did his experiment? That is the dihybrid cross experiment. The answer is yes. What did he find? Law of independent assortment. And to make it very easy, let me not read that up. Let me tell you right over here before we go ahead. Law of independent assortment means that a pair of allele for one character. This is a pair of allele for one character. What is that character? Shape. Round and wrinkled. A pair of allele for one particular character is going to segregate or separate independently of the pair of allele for another character. That is what? Color. So that means that when I say from here one chromosome has to come, it can be capital R, it can be small r. right? And from here when it has to come, it can be capital Y, it can be small y. Is it like, does it say that capital R says that I will only go with capital Y? Is it like that? No, capital R went with capital Y, capital R also went with small y. It is independent. This segregation, who comes from here and who comes from here, is independent of each other. This is known as law of independent assortment. Ma'am, why is this super important? Why are you talking about it right now? Listen, if this was not being followed, you will not find different combination in the gametes. If gametes are not different, you are not going to find difference over here. When you say that new combinations have come, how have they come? They have come because gamete itself is different. Gamete itself is different, that's why you got new combination out there. That is how Mendel came up with the third law of inheritance, which is known as law of independent assortment. And in technical terms, if at all we go, we can just read it out like this. When two pairs of traits are combined in a hybrid, when two traits are being combined in a hybrid, segregation of one pair of character is independent of the other pair of character. It is not at all dependent. Cannot say R round and yellow is going to go. We are going to have a breakup over here. We are going to have a breakup over here. It is never together. Okay, it can be happening like this, it may not happen. There is no guarantee, there's no rule that it has to happen like this itself. Got it? This is the simplest definition, or probably the way I could explain the law of independent assortment. Clear? If clear, just give me thumbs up. You do not have to type anything, because I get to understand. If thumbs up is there, I get to understand that you are understanding. I do not want you guys to waste your time typing up. If you have doubt, you can ask me. No doubt. Law of independent assortment, super clear. And by the way, everyone who is watching this video, do not forget to hit the subscribe button. Clear, right? Akash, da? Akash, I'm a regular student and I'm a teacher. Is it? Oh my God, a teacher meets a fellow teacher. So you're a chota teacher, as in you mean you're a student and you teach? Is it like that, or you're a teacher? So I remember when I was in grade 12th, I used to teach, my juniors out there. So is it like that? That's what I meant, okay? So Akash gave me thumbs up, that means that is also clear. Now I go ahead and over here, let's check out this question. Let's see if at all we are able to do this or not, okay? In a dihybrid cross between a true breeding round and yellow seeded and true breeding wrinkled and green seeded pea plant, the ratio of segregation of round and wrinkled seed trait in F2 generation is going to be what? In a dihybrid cross between true breeding round yellow and true breeding wrinkled and green seed, that means true breeding means it looks like this, round and yellow and wrinkled and green. You have been asked the ratio of segregation of round and wrinkled seed trait in F2 is. Round and wrinkled seed trait in F2 generation is going to be. Any guesses for the answer? You have few options: 9:1, 3:1, 9:3, 3:3. Any guesses, my dear children? What will be the answer for this? They're not asking about the entire thing, they're just talking about round and wrinkled. Do note that. Akash says 3:1 is the answer. That is actually correct. That is actually correct. It is going to be 3:1. Why 3:1? Perfect. Akash, why it is 3:1? The reason being that, just imagine this like this, okay? So round and yellow, forget about this. Capital Y. So doesn't it look like capital T? If at all, I just remove this. Doesn't it look like capital T and small t? Whenever you were crossing them in the next generation, okay, in the round 2, what did you find? 3 of them were tall, 1 of them were dwarf. The same thing happens over here. When you cross this and this, when you're talking about just this and this character, only one particular character, 2 different traits, you have to follow the monohybrid ratio. It is going to follow the monohybrid cross ratio. It is going to be 3:1. If I'm talking about the phenotype, it's talking about the phenotype only, right? Now over here, listen to me very carefully. If at all I want to find the answer in a different way, I can do that. You have been asked about round and wrinkle, right? I can go like that also. Listen, this is round, this is round. 9 plus 3 is equals to what? 9 plus 3, both of them are going to be round. 9 plus 3 is equals to 12. Wrinkled, 3 plus 1. This is wrinkled, this is wrinkled. So 3 plus 1, 4. And if you take the ratio, it is again going to be 3:1. It is again going to be 3:1, which is why the answer for this question is going to be 3:1. Got it? Got it? Now, sorry ma'am, I am a 2026 KCET student. What's the rank jump in first and the second rounds? Depends on different college, Lochin. I'm so sorry, I do not remember it like that. It totally depends from college to college. And okay, got it? This question I hope is super clear for everyone who's probably watching this video and not answering and probably shy to answer. So round and wrinkled is being asked. It is just going to follow the monohybrid cross issue. Next question is going to be interesting. Okay, try to solve this particular question. A pure breeding pea plant with round and yellow seeds was crossed with a pea plant having wrinkled and green seeds. Wrinkled and green seeds. Again, it's like this. Capital R, capital R, capital Y, and small r, small y, small y. On selfing of the F1 hybrid, we got the F1, okay, we got the F1, it looks like this. On selfing of the F1 hybrid, it was found that 64 progenies were made. Okay, after crossing them, you have got 64 progenies. Now the question is, find out the number of F2 progenies showing non-parental characters. How many of them are going to show the non-parental character? It's not going to be like the parents. Non-parental, if we have 64 progenies, so ma'am, we do not know. In 16 we saw that basically when we were talking about 16, ma'am, you said that out of 16, over here 3 plus 3, 6 of them were non-parental. They were showing non-parental character. How can we talk about 64? That's going to be a big number. Do we cross it and see it? How do I know the answer? I do not know the answer. So what do you think? What do you think could be the answer for this question? Anyone? Do you want to give a wild guess? Any guesses over here? Shall I try? Children, any guesses what could be the answer for this particular question? Yes or no? Probably I'll go ahead then. Okay, I'll go ahead then. Listen to me very carefully. When you talk about all of them and when you say 64 progenies, you can think like this. Okay, we know 9:3:3:1 ratio is there, and we saw that 9 of them were round and yellow like parents. Okay, 1 of them was wrinkled and green like parents. And over here these three were different. This was round, was with always with yellow, this time it was with green. And this was wrinkled, was always with green. This was what? This was with what? Yellow. Right? So 3 plus 3, 6 of them. If I have to find out of 64, what do? I just use this. 6 out of 16. This total was 16. 9 plus 3 plus 3 plus 1 is equal to 16. 6 out of 16 was non-parental. Now we have to find out of 64. So we multiply it by 64. 16 into 4, it becomes what? 16 into 4, 64. 6 into 4, it becomes 24. Out of 64 progenies, 24 of them are going to have non-parental character. Did you understand it? And now suppose if at all you have been asked about how many of them are going to be parental, having parental characters. How do we find that? 9 plus 1, 10 of them are going to be like the parents. Right, the same thing applies over here. 10 by 16 into 64. We get what? 4 into 10, 40 of them. 40 of them are going to be parental, 24 of them are going to be non-parental. 24 plus 40 is equals to what? 64. That is what we searched with. So that is what we started with. So the answer for this question is going to be A. Yes, Akash, it is going to be A. Got it? That is how we solve this question. This is a very important question, and these type of questions could also be coming in KCET examination. So make sure you are very well versed with that. Done? Clear? Done? Shall we move ahead then? Cool. If it's clear, can you give me a thumbs up? If it's clear, can you quickly give me a thumbs up? Only if it's clear, okay? Otherwise, Okay, perfect. Now listen to me very carefully. Okay, so what happened is Mendel did his monohybrid cross, Mendel did his dihybrid cross experiment, and then came so many exceptions. We saw there were people saying, Mendel, you were wrong. You said that A is going to be completely dominant over small a. But we saw a different thing in incomplete dominance, where it was not complete dominant, red and white we crossed, we got pink. Okay, similarly, people went about talking about the blood groups. You told that there were only 2 alleles for one particular capital T, small t, but we are seeing multiple alleles, IA, IB, IO. What about that? Okay, now talking about blood group, 2 alleles were both equally dominant, co-dominance was also there. What are you saying? This was not true, Mendel. Now, the next thing Mendel said, basically one gene codes for one particular character. That was also wrong. Some people said that, how can you say that? One gene can have multiple effects. Pleiotropy, we studied about it in the previous session. Then one more thing was there. Mendel also told that one particular gene is responsible for one particular character. But in polygeny, we studied multiple genes are coming to code for one particular character, your skin color, your height and everything. Again, Mendel was wrong. So then, in this particular scenario, everyone went against Mendel. This is something that I am making up, okay? Don't think that people went against Mendel, okay? But anyway, I am telling you, it was very difficult. Even though Mendel did his experiment, he spent 7 long years on something, okay? On experimenting on pea plants. Just imagine how boring that would have been. But still he went on doing, even though there were so many people saying that Mendel, you cannot do it, you did that, Mendel kept on going. That's something that we have to learn from Mendel. But anyways, there were several other reasons also, which is why Mendel's work was not being, properly known to everyone. We know about him, but that was not the case then. Why? What was the problems or the issues that they were facing? First thing, communication was difficult. As of now, if at all I want to talk to Akash, probably I can talk like this. Okay, Akash Macha is there, I can just probably type a message in the chat session and probably talk to him, or talk like this. Communication is not difficult now, but communication was difficult then. Remember, he was doing his experiment in 1856 to 1863, very long back. So, even if he identified something, basically he could not tell it to anyone that I found this, please do further research about it, I know only this much. He was not able to say that. Second reason, his idea of discrete factors and no blending was not accepted. That means this and this allele is there, they are not going to blend, you are not going to have a medium character. Okay, this was not accepted because people saw it. People saw that blending was happening in incomplete dominance. You got red and white pink color, right? Now using mathematics in biology was unusual and not accepted. So initially everyone was praising him for using mathematics in biology, but later on people were like, why are you using mathematics in biology? Use biology in biology, don't use mathematics. Does not make any sense. Now that does not make any sense to us also, but yes. Okay, next thing, he had no physical proof of what genes were or how they looked. So he was the person who said that factors they move, okay, into gametes. One factor is going to go from a pair of factors. So, but he didn't have a physical proof. He could not show where are these factors located, how does it look, and things like that. So which is why he could not show? The reason being that he did not have papa person, he did not have microscope back then. So these were the few things that went really wrong, and we thought that downfall was there for Mendel's. Okay, we thought that he had a downfall in his life. Similar to us, we also have a downfall. Today we are super great, we might be scoring really good. The next day probably our scores might not be great, we might have fought with our friend, or something might have happened at home. Okay, we all have certain things in our life. For him also it was there. But after everything comes a day where everything is going to be back to normal. Again he will have a downfall. This is how it goes. So in Mendel's life also it happened. And he just imagines, for 7 years he worked, but only after 37 years, okay, there came a twist. There came a twist in his life, but only after 37 years. What was this twist about? There were 3 scientists, okay, remember their names. Correns, Shemark, as well as De Vries. Correns, Shemark, as well as De Vries. I'm going to show their spelling and everything, do not be worried. These 3 people came in 1900. After 37 years, in 1900, they came and they were experimenting on the pea plants and things like that. So they had found a similar type of experiment. They got the results and they went ahead and searched that someone else also do the same. Then they found that Mendel also someday was doing this experiment. Mendel was also somehow doing this particular experiment and he got the exact same results. So basically we say that there was a rediscovery of Mendel's work in the year 1900. This is a very important KCET question. This is a very important KCET PYQ. So there were 3 people who were there. One was was Correns, the other one was Erich von Schemark, and the last one was Hugo de Vries. Okay, Correns, Schemark as well as de Vries, three of them, they came and they rediscovered Mendel's work in the year 1900. Now at this particular point of time, microscope was there. People were able to go within the nucleus and see that there was chromosome, colored bodies and things like that. Okay, so just after this, after 2 years of rediscovery of Mendel's work, basically two more scientists came added up. It was Walter Sutton and Theodore Boveri. It was Walter Sutton and Theodore Boveri, and he said that both of them said that, you know what, I can see the chromosomes right now under the microscope, and the behavior of chromosomes and the behavior of the factors that Mendel was talking about seems to be the same. That means, in simple language, what was Mendel saying? That whenever, suppose this is a parent, whenever they are forming the gamete, one capital T goes over here, and the pair of factors are going to separate, and one of them goes to each gamete. Right? This is what Mendel was saying. And the same thing was happening up here also. If at all we see, according to them, whenever they were seeing one chromosome goes to one particular gamete. One chromosome was going to one particular gamete during the process of gamete formation. This is exactly what they saw in the terms of chromosome. This is exactly what they were seeing in the terms of what factors that Mendel was talking about. So they were like, what if these factors that he was talking about are there on the chromosome? What if these factors that Mendel was talking about are there on the chromosome? So whenever the chromosomes are going to separate, that's when the factors are also going to separate. Exactly. And that was found out by what? These two people, Walter Sutton and Theodore Boveri. When they found it out, they gave a theory that was known as chromosomal theory of inheritance. They said that pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors that they carried. For example, right now, when I have capital T and small t in the gamete, okay, this is a gamete and this is a gamete. When I say that they are fusing, basically I mean that the chromosomes are fusing together. The chromosomes are fusing together. When I say they are going to separate, I mean the chromosomes are going to separate from each other. So pairing and segregation of a pair of chromosome is just going to be working parallelly with the segregation of the pair of factors that they carried. This was chromosomal theory of inheritance, and for this obviously they had to use a lot of brain around it. Are you understanding it until this particular point? Clear? If clear, can you give me a thumbs up? If you have understood that three scientists came up first, who are those? Correns, okay, Schemark, as well as the other person. Correns, Schemark, and the other person, who was that? Hugo de Vries. These three people came together for the rediscovery of Mendel's work in the year 1900. Then after that, okay, they said that all what Mendel was saying was always correct. Now, after 2 years, in 1902, two people more came in, Sutton and Boveri. Okay, they said that basically whenever chromosomes were segregating, factors was also segregating. That means probably these chromosomes were having those factors, or the factors were there on the chromosome itself. This is known as chromosomal theory of inheritance. Okay, works. Pawan, hi ma'am. Hello Pawan, how are you? Extremely sorry I'm so late. It's totally fine. I'm glad that you're here. Okay, so I hope this part is clear. Now, if it's clear, thumbs up, Macha. I'm waiting for your thumbs up. Give me thumbs up, da. I'm not asking for giving me your property or gold or anything. Just give me one thumbs up so that I understand that children are understanding. No? Drink water, by the way. Okay, so no one is still interested in giving me thumbs up, which is why probably I would have to go ahead with the next thing. So listen to me very carefully. Whenever people come and say, for example, tomorrow Jasmine Ma'am comes and say that I have found this particular thing, will you believe me? Will any one of you believe me? The answer is no, you will not believe me. Why will you believe me? You will ask, give us proof. Give us proof, then we will believe. Yeah, Akash gave me thumbs up. Thanks, Macha. Okay. So basically, everyone wants a proof. Whenever you say something, you need proof. So for chromosomal theory of inheritance also, there was a proof that was needed. And that is the proof that we're going to talk about right now. It was done by some other person. We're going to see him. We're going to hear about him. It's a very important part. Do not miss it out. But before I go ahead with that, there's one important question. Which three scientists independently rediscovered Mendel's work? Can you guess the answer? A, B, C, D. You have got all the options. From here you can tell which three scientists, who were those three scientists out there who independently rediscovered Mendel's work. Any idea? D. D is what Pawan is saying. D. Okay, Pawan is saying D. So we have to go with D. D is what? De Vries, Correns as well as Schamak. Exactly. These people independently rediscovered Mendel's work in the year 1900. Please do not forget that. Akash says B. Sutton, Morgan and Bridges. No, Baba. That was not Sutton, Morgan and Bridges. I hope you have got it. The answer for this question is going to be D. Three scientists. Remember? These three scientists we were talking about, look at them carefully, these three people. Okay, Correns, Schamak as well as De Vries. Okay, yes. Now, we're going to go ahead and we're going to see the proof of chromosomal theory of inheritance. Okay, someone had to prove it. And basically, it was none other than Thomas Hunt Morgan. So he was hunting, okay, he was hunting when he went to hunt. That's when he found a unique organism, for which he thought that I have to experiment on this organism and prove the chromosomal theory of inheritance. Ha ha ha. So that's not the thing. Okay, Thomas Hunt Morgan was sitting ideally one day and he saw in front of his eyes fruit flies were moving around. So he thought that I should be experimenting on them. Even that's not true. But listen to me. Fruit flies is something that he was working around. You also call them as Drosophila melanogaster. What do you call it? Drosophila melanogaster. They look like this. Okay, they're cute little organisms out there. They are coming around. If at all you keep your food open, probably they might be coming around, sitting out there, eating some food and going up. Okay, so that is how usually they work. Now, why out of all the organism out there, Thomas Hunt Morgan chose Drosophila melanogaster for his experiment? First thing, okay, Akash gave me the answer D. Thanks, Da. Okay, so the first thing is that they could grow on a simple medium. They didn't ask for burger or pizza or anything. They said that we can grow on simple medium, and that is very, very important because you are going to do the experiment so many times, so you cannot afford a very, very expensive medium out there. Second thing, short life cycle. That means I do not have to wait for years to see the results. Within 2 weeks, I could easily spot my results out there. So this was very important. Next thing, whenever meeting was happening, they produce so many progenies. Now when I get so many progenies, I can probably experiment more on them. So that was another reason. High progeny. This is a second PUC question, by the way. Okay, next thing, clear male and female distinction. So for mating to happen, or if at all we want to do it artificially also, we need to understand the male and females, and in organism it's very difficult to find out. But here these guys had a clear male and female distinction out there. Okay, usually the smaller one is going to be the male and the larger one is going to be the female. So this is a female. Look at this. Okay, it's what's big, and this one is small. This is going to be a male. This is a very unique feature. Next thing, visible hereditary variation under a low power microscope. For example, I want to, they have some variations out there. Some people have red eye, some have yellow eye, white eye. So many different things are there, right? In this particular case, if at all I want to see those variation, I do not require a high power microscope out there. Rather, it is clearly visible under a low power microscope also. It was clearly visible under a low power microscope as well, which is why we thought that Drosophila melanogaster, that's it. You are my guy. I am going to do the experiment on you. That is why Thomas Hunt Morgan went with Drosophila melanogaster. Is this clear to you why he chose melanogaster? And this is a 3-marker question in your second PUC. Make sure you know all the features. Okay, if clear, give me a thumbs up. If it's super clear to you, can you quickly give me a thumbs up? Motivational is also here. Hello. Perfect. Why I'm getting that translate option out there? Is there some issue with my phone? Okay. It is known as sexual dimorphism. Exactly, it is known as sexual dimorphism. Okay, it is known as sexual dimorphism. What do you mean by sexual dimorphism? Dimorphism means 2 type of morphology. So here, in the case of male, it has a different morphology, external features. In the case of females, it is going to be having a different feature. Okay, yes, correct. And Pawan and Akash, thanks for giving the thumbs up. Done. Now the class is banger. Okay, now This was Thomas Hunt Morgan, by the way. He's looking at you guys only, okay? So did he find something else? Let's check it out. So he found something very important, and it is known as, what do you call this as? Linkage and recombination. And I am pretty sure this particular term might be eating your brain somewhere here and there, and probably this particular picture also could be eating up your brain somewhere here and there. If at all any of this is basically making you sad, then probably I'll tell you it's not important to study the entire picture. There are only few things that you need to know, and I hope we are ready to go ahead with this. If at all you listen to me, I promise you will never have any confusion around linkage and recombination. So Baba, just assume this is a chromosome that we have, and this is the chromosome that we have. Cool? 2 chromosomes. Okay, and here also I have 2 such chromosomes, one chromosome and the other chromosome. Here I have a gene, that is A gene. Here I have a gene, that is B gene. Here I have a gene, that is small a gene. Here I have a gene, that is small b gene. Okay, I have small b gene over here. Here I have capital A gene, here I have capital B gene, here I have small a gene, here I have small b gene. So listen to me. Whenever you're talking about organisms, Baba, they are going to undergo sexual reproduction, and for that they have to form gametes. Now for forming gametes, you need meiosis to happen. Lot of things, different things are there. Okay. So while this whole process of division and everything happens, there is one more process that happens along with it. You call this process as, any guesses on what this process is known as? What is going to happen over here? Any guesses what happens over here? I'll tell you. It's known as crossing over. It's known as crossing over. This happens every time. Whenever you make the gametes, the chromosomes over here, when they lie close to each other, no, they put their legs around each other, then sleep. So usually when you sleep, I sleep like that. Okay, I put legs around the people I am with, and basically that's how I sleep. So that is exactly what is happening over here also. Okay, here also the same thing is happening. So when you have chromosome like this, they're going to swap. They're going to overlap at an area and basically swap their things. They are going to, yes, Pawan, it is known as crossing over. They are going to swap their things. Now just suppose, in that particular condition, if that is happening, and that is happening over here, below this, what are you going to see? What are you going to see? You are going to see some unique things out there. This part goes this side and this part goes this side. So if I really want to make a chromosome, a new chromosome, it is going to look something like this. I have capital A over here, and basically I would be having, instead of capital B, I will be having small b. Why? Because it got interchanged. Similarly, over here, I will be having small a, and here, instead of small b, I would be having what? Capital B. Oh my God, ma'am, that's crazy. That looks so different from this one. That looks so different from this one. So what do I say? I say that here I have got a new combination. And a new combination, basically I say it's a recombinant. It's a recombinant that is being formed, and the percentage of recombination over here, there is more amount of recombination that I found over here. But how do you understand whether it's more or less? If at all you do not compare, so we're going to compare. We're going to see what is going to happen over here. The same thing happens over here. Let's have a thing. Over here it moves this side, over here it moves this side, the lower portion. Now when I do that, what do I see over here is that, okay, what do I see over here is, the lower portion got exchanged, but the top portion is still there. Capital A is still with capital B. Small a is still with small b. Here, do you find recombination? Here, do you see recombination? Yes, it is there because the lower portion has changed. But do you see recombination among them? What do you say? We say it's very less. The recombination is there because overall, if you're talking about, yes, recombination is there, but here it is going to be very less. Very less amount of new combination is being formed over here. Now, why is this happening? What's the reason for this to happen? Because, Baba, these two genes over here are very far away from each other. They are not linked. Linked means it's basically a tendency of the alleles to stay together and to move together. Here when I see them, they are not tightly linked. They are not too close to each other. I say they are going to be what? Loosely linked. They are going to be loosely linked. And if they are loosely linked, they will show more recombination. If you didn't understand, listen to me. You and someone else. For example, I'll say Okay, I am going to use two people. Okay, Pavan and Akash is there. Pavan and Akash, they are best friends. They are best friends. Okay, from starting point on, from KG1 onwards, they are together. Now, will they allow motivational to also come into this particular frame? They will like, no, we are good. We will be happy like this. So when you're best friends, you will not allow new combination to happen. So that's what I'm saying. Okay, that's what I'm saying. That over here, when you see, they are very close to each other. What do you call them as? Tightly linked genes. You call them as tightly linked genes. These genes will not allow any new combination to happen. But on the other hand, just suppose that Pavan and Akash has met yesterday. Pavan and Akash has just met yesterday. Now they are pretty much okay if at all motivational also comes along. That's not a problem. Why? Because they have just met yesterday. They are fine. They're loosely linked. They're not too close. Which is why they can allow more combination to happen. That is exactly what is happening over here in this particular case. Is it clear? So loosely linked genes over here will show more recombination. Tightly linked genes over here are going to show less recombination. And the maximum percentage of recombination that everyone has is basically 50. This is the maximum. This is going to be maximum. Any number close to 50 will be showing high amount of recombination. Any number which is far away from 50, for example 1 and all, the recombination is going to be less. Is this particular thing clear to all of you? If yes, give me thumbs up. I'm going to go ahead. I'm going to talk about that particular picture also. Loosely linked genes, more recombination, less parental combination. Exactly. More recombination means new combinations are there. Parental combinations will not be seen much. Okay, parental features are not going to be seen much. Perfect, Pavan. You got it. Okay? Cool. Tightly linked genes, less recombination and more parental combination. Perfect. Got it. That is sorted. Yes, ma'am, got it. Akash also got it. Motivational dear, I hope you have also got it. Okay, now listen to me carefully. Here in this particular case, there is another feature with which the help of which they are going to explain. They are going to explain. So I'm just going to remove this portion over here such that, just a second, I get a little space to draw certain things. Okay, now listen. So there are few genes over here. Over here, I am saying there are 3 genes, and you have to tell me what is the position of them going to be like. So I am saying that there is yellow body genes, genes which is responsible for making the body of the organism yellow, and there is white eye gene which is responsible for making the organism's color white eye, so eye color white in color, and basically there is one more gene that is miniature wing. So in that case, the organism is going to have small wings. And these 3 genes are there. These 3 genes are there. I am telling you, yellow body. Okay, yellow body. Okay, now I cannot take that. Can I break in 30 minutes? Yes, dear Akash, yes. Okay, listen. So I cannot use that funda over here. So anyways, I'll go directly. So listen, there is a gene over here. There is yellow body. Okay, I'll give a question later on. And there is a gene over here that is white eye. Okay, and there is a gene over here that is for miniature wing. So you always remember that yellow body, and in yellow body you have white eye. Okay, you're going to be white eye, and then this particular gene which is miniature wing is going to give small wings out there. Okay, now I am saying that he found one very important thing. He found that the white eye and the miniature wing gene showed 37.2% recombination. Whereas yellow body and white eye showed 1.3% recombination. Why so? The reason being is very simple. Baba, this is here, they're very far from each other, which is why if at all they do crossing over, this guy goes off, this guy moves to the other chromosome, which is why we say the percentage of recombination is what? 37.2. It's very close to 50. It is very close to 50, so that means it is more recombination out there. Why? Because it is, if you're asked why, then it is because it is loosely linked. It is going to be loosely linked. On the other hand, when you look at this, yellow and the white eye, over here it shows 1.3% recombination. Why? Because this is going to be what? Tightly linked. It is going to be tightly linked. That is the reason, and that is something that you have to study. You need to remember yellow, white, and YWM. Okay, YWM, this is the order that is there. And here they show white and miniature wings show 37.2% recombination. Yellow and W, okay, sorry, Y and W, both of them in alphabetic order also, they are very close, you can remember like that. Okay, they show 1.3% recombination. Very, very important. Yes, Pawan, perfect. Now, can I ask you a question? Okay, since I already spoke about all of it, I think I can also ask you a question. I am giving a question which goes like this. I am saying there are 3 genes. Okay, there are 3 genes, A, B and C. Okay, I am saying that A and B shows 35% recombination. A and B show 35% recombination. Okay, A and C shows 5% recombination and B and C shows 30% recombination. How is the order of arrangement, or what is the order of arrangement? Which one comes first, then comes what, then comes what? Any guesses? You have to tell the order of arrangement. 1, 2, 3. Pattern will be ACB. How? It's actually correct. So listen, for children who do not know, who didn't get it, let me tell you. So it goes like this. Listen. If I make A and B has the highest, so we have to spot the highest one. Highest is 35%, so I will make it distance. Okay, I will put it a distance 35%. Now, the second highest is what? B and C. B and C I do not have. So see, if at all I place it over here, is it right? Then this has to be closer. What is this? It shows 5%. So it is actually good. It can be here, it shows 5%, and rest, this is going to be what? 30. So 30 plus 5, it becomes 35. So what is the arrangement? A comes first, then comes C, and then comes B. This is how it goes. Pattern will be ACB. Perfecto. Got it? It's so simple. So if you know the percentage of recombination, basically the answer also becomes super easy for us to find out. Cool. Good one. Children, everyone, motivational, where are you? Akash? Panto gave me answer. So yes, that is how it goes, and that is exactly what is written in the Morgan's experiment. He has mentioned this particular thing, that is, genes on the same chromosome tend to be inherited together. This is known as linkage. Genes which are there on the same chromosome, they usually move together. This is known as linkage, but the strength of linkage varies depending on the distance between the genes. There are tightly linked genes, they are going to be very close on the chromosome, and they are going to show very less recombination. Why? Because they always moved up together. Example, white eye and yellow genes. White eye and yellow genes, they show 1.3% recombination, which means they are tightly linked. Loosely linked genes, they are farther apart on the chromosome, higher recombination. Example is white and miniature wing genes, they show 37.2% recombination. Okay, so the closer the gene, the stronger the linkage and lesser the recombination. That is the basic rule that we have over here, and this is very, very important. Now, have questions come from here? Obviously, second PUC gives so many questions from this particular topic, so you should not be missing it. Similarly, KCET also has given you the question. I'm going to show you that. But before I go ahead, there is one very important thing that I want to share. So this was the teacher, Thomas Hunt Morgan. You know what? Thomas Hunt Morgan also had a student like this, just like me, having students like you. Okay, his name was Alfred Sturtevant, and he did something such that the teacher shocked and the student basically rocked it. So what was it? What was that Alfred Sturtevant did? Basically, he said that, sir, you gave us the percentage of recombination, that this is the percentage, this is a percentage between recombination between two different genes. With the percentage of recombination, I can easily locate the genes. That is what exactly we did. A, C and B, we got the percentage of recombination. Okay, then we were able to locate them. On the basis of that only, we were able to find the pattern on the basis of that. This process is known as what? Gene mapping. This is what you call as gene mapping. So Alfred Sturtevant used the frequency of recombination. He used his, search data for that between gene pairs on the same chromosome as a measure of distance between genes and mapped their location. So I hope one day probably you guys also would be making me proud. One day you guys also will be shocking me for a good reason, with a good reason. Right? Is this clear to you? This is all about linkage and recombination, nothing else. We're going to solve some questions and we're going to cement it in our brain. If it's clear, can you quickly give me a thumbs up? Perfect. Thank you so much, Pawan. Okay, the next thing that we have is a question. In Morgan's experiment with Drosophila, this is 2019 KCET question. In Morgan's experiment with Drosophila, when yellow-bodied, white-eyed female was crossed with brown-bodied, red-eyed male and their F1 progeny were intercrossed, what was the percentage of recombinants in F2 generation? Now, when someone reads it, they are like, oh my God, what am I reading? So listen to me carefully. Read it once more. In Morgan's experiment with Drosophila, when yellow body we have studied, okay, white eye also is something that we have studied, was crossed with brown body and red eye. This and all we did not know. Yellow and white we saw, and we have been asked about recombinants in F2 generation. So the answer is going to be simple. You just have to think about yellow and the white eye. What do you think will be the answer? Yellow and the white eye, are they close to each other? Do they show very much amount of recombination, or do they show very less amount of recombination? What do you think? D, ma'am. Yes, it is going, Dia. Dia. Yellow and white. Y and W. Both of them, they are similar. They are together in the alphabetic order. They are together in the alphabetic order. Think, Pavan. Once more. Yellow and W. How much percentage? Can it be more? It cannot be more. It cannot be 37.2, right? It is going to be what? Oh, yes, it is C. Exactly, it is going to be C. 1.3 percentage. A, B, C, D, E, F, G, H, I, J, K, L, M, N, P, Q, R, S, T, U, V, W, U, V, W, X, Y, and Z. Okay. No, I cannot use my formula over here. So W and Y has one guy in between, so you remember 1. And 3 though, okay, you have to anyway remember. So I was trying to make any trick out there but didn't work. Sorry, I failed utterly. Anyways, the answer for this question is going to be 1.3 percentage, and I hope you know why. Right, you have got that particular reason why it is 1.3 percentage. Now there is one more question. Don't go. One more question is there, and that is this. This is a 2023 question. Okay, gene mapping technology was developed by Correns, Sturtevant, Mendel, and Schemark. Who was the person who came up with gene mapping technology? Gene mapping means locating genes on the chromosome. Who was the person who came up with that gene mapping technology? B. Oh my God, Nainika is there. Hello, Nainika. And Nainika came up with a banger. Say that it's B, Sturtevant. Exactly. It is B, Sturtevant. His actual name is going to be what? His actual name is going to be Alfred Sturtevant. And Pawan also gave B as the answer first. Perfect. It's going to be Alfred Sturtevant, and this is what we spoke about right here. Right? Teacher shocked, student shocked. Why? Because he came up with, the student, Alfred Sturtevant, came up with gene mapping. Okay, now this is second PUC question which is there, okay, which has come before. That is, recombinants obtained in both Mendelian and Morgan's experiment, yet the process involved are different. Comment. So how do you give the answer for this? Is a 2-marker question. Recombinants are found in Mendel's experiment, in Morgan's experiment. How it is different? How do you get new amount of recombinants out there? What are the processes? So Morgan's experiment is something that we saw. Here you get recombination because of what? Crossing over. Okay, the chromosomes were crossing over. That is why you get recombination. We know that. Okay, we studied about that right now. In the case of Mendel's crosses, why did we get recombinants? Because we studied that basically, if at all over here I have capital R, small r, and I have capital Y, small y, in an organism which looks like this. And when I cross it, basically I get to see multiple type of gametes. Capital R sometimes goes with capital Y. Capital R sometimes goes with small y, small r sometimes goes with capital Y, small r goes with small y also. You have multiple type of gametes, and when you have multiple gametes, they are also going to fuse to form multiple type of progenies, right? So what is the reason? This is law of independent assortment. We studied in Mendel's crosses. here itself, in this class itself, that law of independent assortment is being followed. From a pair of allele, anyone can come independently of what is coming from here. It does not say, capital R does not say I will only go with capital Y. Okay, it can go with small y also. There can be multiple combination the gametes, which is why you get multiple progenies, multiple type of progenies as well. So always remember, in Mendel's experiment, it's because of independent assortment. In Morgan's experiment, it's because of crossing over process. Okay, is this clear? We have studied it, which is why I'm not investing too much time into this. Is it clear, or do you want me to go a little bit, in detail and explain you guys? If it's clear, can you give me a thumbs up? Here, okay. By thumbs up, I mean here, okay. If it's super clear to you, I would not mind seeing a thumbnail. No, it's not clear. Okay, yes ma'am, understood. Perfect. Let's go ahead. Now I might look funny from back, but it's okay. Okay, differentiate between linkage and recombination. That is the next question that we have over here. What is linkage and what is recombination? Linkage is what? Tendency. Or for example, I am linkage. I want both of you guys to move together only. Okay, linkage means the tendency of genes located close together in the same chromosome to be inherited together. In every other chromosome or any other gamete it goes, basically it has to move together only. These two genes, the tendency of the genes located close to each other to move together, to be inherited together, that is what you mean by linkage. Okay, what do you mean by recombination? It is a process by which we get new combinations of genes. That is what you mean by recombination. Now you tell me the next difference that is there, it can be on the basis of who brings more variation. So recombination definitely brings more genetic variations, okay, because you're talking about new combinations being made up there. Linkage reduces the chances of more genetic variation and maintains what? The parental character only. It wants to maintain the parental character itself, no new characters. Okay, so if at all you give these 2 points, you can get 2 marks very easily. This is also a second PUC previous year model question paper question. Got it? Fine. So now we are going to go ahead. Yes, per one. Thank you. Next, what are the conclusions drawn by T.H. Morgan's experiment over here? He said that higher the linkage, lesser the recombination, or we can say more tightly linked genes, less recombination. Okay, if the genes are loosely linked, they will show more recombination. Fine, this is what we studied. We studied about it so many times, which is why I do not want to spend time. This is again a 2-marker question. Now, after we have finished, one more question is there. T.H. Morgan selected fruit fly for his genetical experiments. Why? What was the reason for it? This also we studied right now, but we are going to quickly revise because this is a longer thing, right? So it might need revision. So listen to me. First thing, if I am a Drosophila, I do not need burger, pizza, anything. I can grow in a simple medium. So that was the first thing. Second reason, they have a very short life cycle. Okay, they will only survive for 2 weeks. So basically, if at all I want to see the results, I can see within 2 weeks itself for my experiments. Third thing, whenever mating happens, what is going to happen? There are so many progenies being produced. If there are so many progenies, I can work around so many progenies also, right? The next thing, basically, they are going to show sexual dimorphism. The male and the female is easily distinguishable. The males are going to be smaller and the females are going to be larger in size. This is known as sexual dimorphism. 2 different sexes will be 2 different in appearance as well. Okay, and the last point is, if at all we are looking for any type of variation, yellow body, white eye, blah blah blah, anything which is there, it can be easily seen with the help of a low power microscope. I did not need to bring a very big microscope, electron microscope, so when a big microscope is not needed over here, which is exactly why I could use Drosophila melanogaster for the experiment. So this is again a question. This was actually a 5-marker question. In 5-marker there is A and there was B. This was the A part, which contained 3-marker for this. Okay, I clearly remember this. This was also there in the previous year. Cool? Clear? Sorted? Okay, now we are going to go ahead with the next topic, and that is sex determination, which is going to be the last topic for the day, and I hope I finish it at the correct time. By the way, every one of you watching this video, I hope all of you have subscribed to the channel, and motivational is back. Yes, so sex determination is going to be super fun. Okay, it's one of the most easiest thing to learn. So let's go one by one. I'm going to use this particular slide for that. Okay, let's go. So over here, first of all, there was a guy whose name was Hen King. What was his name? Hen King. Funny name, right? But yes, Henking found that insect, he was studying insect, basically grasshopper and things like that. He found that the sperms of the insect, few of the sperm, they carried X chromosome. Few of the sperm, they didn't carry any sex chromosome. He was like, what is happening over here? So he went on researching about it, and then he came to conclusion that basically insects produce two types of sperm. Some sperms carry X chromosome, some sperms do not carry any of the chromosome. So here what is exactly happening is that if I am talking about a grasshopper, the females, they have two X chromosome. What about the male? The male have one X chromosome, and there is nothing. There is no other pair over here. Okay, there is nothing which is denoted by 0. This is denoted by naught. Okay, now suppose when I make the gametes, this is one gamete, this is another gamete. When you make the gamete, the chromosomes separate, one goes to each gamete. The females are going to produce the same type of chromosome only. What about the males? The males are going to produce either the X chromosome over here, or probably have nothing inside the gamete. Okay, now suppose this and this comes together. Of egg carrying X and a male gamete carrying X comes together, it becomes XX. It becomes what? Female. Suppose this and this comes together, X and nothing comes together, that means it is going to be what? Male. Suppose this and this comes together, XX, again it is going to be female. When this and this comes together, XO, this is going to be male. What do you see over here? What do you see over here? That, ma'am, males are producing two different type of gametes. Okay, and females are producing only one type of gametes. Okay, got it. I do not know the term for it as of now, but let's go ahead. Let's go ahead and let's talk further. What happens in our case, human beings? What is seen in the case of human beings? In human beings, Baba, we have XX chromosome and then we have XY chromosome. Boys, that is men, male have XY and female over here have XX. Now what will they give to the gamete? They do not have anything much, it's just the X chromosome, so it will be same type of chromosome that I give, no partiality. In the case of male, some sperm will be carrying X, 50% of the other sperm will carry Y. Suppose this and this comes together, I get XX. It is going to be what? Female. Suppose this and this comes together, it is going to be XY. It is going to be what? It is going to be male, ma'am. XY to male, right? This and this comes together, XX. Again, it is going to be what? Female. This and this comes together, XY. This is going to be what? This is going to be male. 50% of them male and 50% of them female. Okay, ma'am, in both these cases I see one thing, that is males are producing two different type of gametes. And motivational game be the answer. This is known as heterogamy. Exactly. This particular process where you have producing two type of gametes, this is known as heterogamety. Okay, what do you call it? Heterogamety. Okay, two. Hetero means other. Two different types. Hetero means different types of gamete. Now here is male producing it or female producing it? Males are producing it, which is why you call this as what? Male heterogamety. Male heterogamety. Where do you see that, ma'am? Usually in the case of insects, but in your NCERT one organism is given, and that is what? Grasshopper. Grasshopper has something like this. Okay, what about human beings? Humans. And not just humans. One more example is given in your NCERT, which shows this type of heterogamety. Any idea? Apart from humans, someone else also have XY type of sex determination. Guess what? Who is that organism? Anyone can guess. Anyone are freely allowed to guess. Waiting. One more organism shows XY. Motivational say Drosophila. Exactly. Drosophila, you remember? Thomas and Morgan, Drosophila. So human beings and Drosophila, both of them show this type of heterogamety. This is known as male heterogamety by XY type. XY type. Now, there is one question. Ma'am, the females never produce two different type of gametes? Pavan, Drosophila, perfect. Nanik also said something, but because I'm sorry, I did not see it, dear. Okay, anyways, does females also have something like this? Females also want to show heterogamety. Okay, let's see that. Okay, let's see that over here. Here. I was going to make a diagram, but then I thought that's a bad decision. So let's come back. So in this box, I am going to write it. ZZZW. This is a female and this is going to be a male. Okay, over here, in one gamete Z, in another gamete also Z. This is males are producing ZZ, same type of gametes. Here in one gamete Z, in another gamete W. In the females, they are doing so. Okay. Now, when I cross this and this, I get ZZ, that is a male. When I cross this and this, I get ZW, that is going to be a female. When I cross this and this, ZZ, that is going to be a male. And finally, when I cross this and this, I get ZW, that is going to be a female. Do you know in which organism do you see this? Pawan said that birds show female heterogamity. Exactly. This is seen in the case of birds. This is what you see in the case of birds according to your NCERT. Now listen to me very, very carefully. W over here for women. W over here for women. That is how you can remember if at all you want. If at all they give you any question that confuses, my dear children, probably you can use this logic. Z, 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 W, W for females. Okay, this is found in the case of birds. These are the two different things that I wanted to tell. Apart from that, there is one more thing also. We are going to come to that. Okay, so there is male heterogamety where males are producing two different type of gametes. XX, XY type in Drosophila and human beings. XO type in the case of, what do you say, grasshopper. Female heterogamety is also there where females are producing two different type of gametes. ZW in the case of birds. Now one more thing is given in your NCERT. Okay, and that is basically the cutest little organism out there. Not this one, that is also cute by the way, but this one. Ma'am, what are you saying cute? Have you ever been stung by bee? I know, but you know what, their life is very, very different. Okay, if at all you ever get a chance to study about them, I have told that in another video also, go just look out their story. It's really nice. So you know, they have this societal caste system. Okay, there will be a queen, but there is no queen. Okay, there are workers, there are drones, there are so many other organisms up there, and we're going to study about that. That's actually very funny. Okay, it's actually very funny if at all you go around them, but it's very, very nice. So we're going to see their sex determination. This is kind of a little different from the usual ones, and I need some space to make that. So where do I make? If you don't mind, can I just remove it, because anyway you have it over here. You have all of them over here. If you don't mind, can I just erase it off? Because otherwise I will need a new slide. I cannot add a new slide right now. If it's okay, can you give me thumbs up if I can just erase that one? Yes, ma'am. Okay, I became mom also. Okay, nice. I'm happy. So listen. So listen to all my cutest little children. Very, very important. Okay, you have honeybee. Honeybee. In honeybee, we have total number of chromosomes to be 32. How many number of chromosomes do we have in total? Any idea how many number of chromosomes do we have? Human beings have in total? You can guess around. How many chromosomes do we have? Okay, how many chromosomes does Pawan have? How many chromosomes does Nainika has? Motivational has 23 pairs of 46 in number. Pawan, perfect. Motivational says 46, exactly. So we have 46. Just like that. If at all I'm talking about both the set of chromosomes, 23 come together, that's how we get 46. That is two sets of chromosome, right? Over here, when two sets of chromosome combine, you get 32. So this is the highest number. I'll say this is diploid. This is the diploid, or basically 2n. Okay, double set of chromosomes which are there. Then you have one more thing, that is haploid. That is haploid, half of it. It's known as n. Half of 2n is n, and basically here the half of 32 will be 16. So here Nainika said 46, perfect, Macha. Okay. Now look at this. Okay, you have females and you have a male. Female is always having 32 number of chromosomes. That means it is going to be diploid in nature. What about males? They always have half the number of chromosomes. That means they are always going to be haploid in nature. Now suppose I want to make the gametes. What do I do? If I want to make gametes, what division do I undergo? If we want to make sure we want to make a gamete, what type of division do we undergo? Meiosis or mitosis? Meiosis or mitosis? Meiosis. Okay, let's do meiosis over here. I'm doing meiosis. Okay, I'm doing meiosis. I'm not making all the stages of meiosis. At the end you get half the number of chromosomes, right? So I'm making half the number of chromosomes. These are going to be what? Gametes. 32 became 16. Now here this male guy is already having 60 number of chromosomes. Nainika also said meiosis. Okay, and motivation also said meiosis. Okay, now here Baba, when I have 60 number of chromosomes in the case of male, here again meiosis is going to happen, yes or no? Here, meiosis is going to happen, yes or no? What do you think? You can just say yes or no, whatever is correct, whatever feels correct to you guys. Don't look at the book. It's okay, even if you give me wrong answers here. Male is there, already has 16. No ma'am, mitosis takes place here. Is it? I don't think so. I don't think so. Motivational says no ma'am. Okay, no. Everyone says no? Okay then, let's go ahead with no. So actually, mitosis takes place here. Why? Because we cannot reduce it more. We cannot reduce it more. Mitosis means the cell divides, the number of chromosomes remains the same. The number of chromosomes is going to remain the same. So here the cell divided. Suppose you got 2 cells over here. But the number of chromosomes is 16. Now listen to me. This is an egg, this is a sperm. Right, the male and the female gamete. When both of them they come together, 16 plus 16 or n plus n, it becomes what? N plus n, it becomes what? 2n. That means 16 plus 16, it becomes 32. Is this going to be a female or it is going to be a male? This one is going to be a female or is it going to be a male? What do you think? A female or a male? Female. Okay, Nanika, Pavan, everyone says female. So let me write female. Okay, now how do we make the male then? Because male is only 16, no? It cannot fuse with anything else. This gamete cannot fuse with anything else. That gamete also cannot fuse. It becomes 32 then. Then what do you do? Ma'am, then only one method is left. Don't do fertilization. Array, without fertilization, how can we do? So if at all you remember, in sexual reproduction and flowering plants, without fertilization, we made the ovary convert into fruit. So here also that can happen without fertilization. No fertilization is needed. Without fertilization, just with a process that is known as parthenocarpy. No, parthenocarpy is something that we studied in sexual reproduction flowering plants. This is going to be parthenogenesis. Always remember, not parthenocarpy. This is going to be parthenogenesis, with the help of which this gamete, okay, this gamete which is having 16 converts into a male organism. It converts into what? A male organism. And what do you call the male? First of all, how much number of chromosome? Only n. It is going to be haploid in nature. Only one set of chromosome will be there. It will be haploid in nature. Now, I want to ask you a question. Male come from unfertilized eggs and this process called parthenogenesis, ma'am. Exactly. Q&A, what are you saying? Motivational? Sorry, I didn't get you. Okay, anyways, I'm going to ask you a question. Does this son have a father? Does this son have a father? Yes or no? This particular male child which is being formed over here, does it have a father? No. Nanika says no. Okay, everyone allied? Okay, just a second. Okay, Pavan also says no. But male is not possible involved. How is this possible? This is possible. Again, just like how we had, as I said, in parthenogenesis to happen, you do not need gametes. You do not need any fusion of gametes to happen. If there is no fusion happening, it will transform into male. Do you know that there are different types of sex determination? I think in 10th class also we studied. In crocodiles and all, if at all they are put in warm temperature, okay, it changes, okay. There are some changes happening, genetic changes happening over here, the chemical changes happening over here, which turns the organism into different sex altogether. If at all the crocodile puts it in an area where it is too much shade and things like that, it will be something else. So that happens. Okay. So over here, no, it does not have a father. Does it have a grandfather? Does it have a grandfather? The son? Okay, motivation already gave me the answer. No, but have a grandfather. Exactly. I'm asking others also. Yes, they will have a grandfather. Why? Because this son has a mother, and the mother will be always formed by the fusion of two different gametes, one coming from each of the parent. So yes, it will have a grandfather. This is a very important line given in your NCERT, that here the grandfather is there, grandson is there. The son which is there is not having father, it will only have grandfather. Similarly, the grandfather who is there, he will not have a son, rather he will have a grandson only. This is a very important thing. Do remember that. that. Okay, it's a tricky thing, that's why I thought I should be explaining it. This process is known as parthenogenesis, where no fertilization happens over here. There's no fusion, which is why you just have half the number of chromosomes. You do not have the panel thing. And one more interesting fact that I want to tell is, this is known as a drone. What do you call this as? Drone. This is a male organism that is formed, is known as drone, and it has only one function, that is to mate with the queen. So you know what, in the society of honeybees, one queen is there in each, okay, in each thing one queen is going to be there. One female turns out to be a queen who is going to get the royal jelly and the royal treatment and stuff like that. This queen who is there, she is going to go ahead with a nuptial flight. She is going to fly above, okay, this is known as nuptial flight. This is for mating. Now all the drones which are going to be formed, the men, okay, they are also going to go up for the flight. They are also going for the mating purpose. Now it goes up really high to check who is able to come up there. It ensures that only the best one basically comes and fuses and gives the gametes. You get it? And best one comes and fuses and make the zygote out there. That is why they move really, really up. And finally the one who reaches, they mate there. Okay, they are going to have the mating process there. Then what happens? Guess what? Any guesses what happens? Any guesses what happens after the mating? In the case of honeybees? I'll tell you, the drone dies. Okay, the drone is going to die. Only for mating purposes right up there. Similarly, okay, when I talk about the other fertilization, ma'am, so fertilization is going to happen for sure. Okay, that is going to be there, but the drone is going to die. That is a general thing that is going to happen. Okay, and when you come back to the honeycomb and things like that, there, you know, so many females are formed, but I told you only one becomes the queen, right? What about the others? They become workers. And in workers also, okay, in workers also, there are different type of workers. Some of them make sure that they go for hunting, well, basically not hunting, they go for search of food, the pollen grains and stuff like that. They are the ones who move out. Okay, there will be some security guards out there who will make sure that no one enters over here and I'll fight back. Okay, there are some people who are going to make sure that take care of the honeycomb, they make the wax and everything out there. That is going to be a different thing. Some people will be there who are going to make sure that the royal chamber where the queen is there is very, very well nice. It has been arranged properly. This is how it goes. That's why I said it is very, very fun. So when I think in my BSc I studied in ethology. This is a very fun topic. If at all you can, go search it in YouTube somewhere, okay? Social organization in the case of honeybees. You might also end up liking it, watching it, so please do so. Anyways, is this concept super clear to all of you? If it is, just give me a thumbs up. If it is, just give me a thumbs up. And if at all you come across some fun fact about honeybees, do let me know in the next session. I'll be waiting. Okay? If this is clear, can you give me a thumbs up? Or if you have any doubts around this, if you have any doubts around this that you want to ask me, you can. Go ahead and ask me. Okay. So children, yes, ma'am, crystal clear. Thank you so much. Thank you so much. Now here is a question that I have. In male heterogametic type of sex determination, what do you find? In male heterogametic type of sex determination, what do you find? This is 2023 question. Female parent produces dissimilar gametes. Male parent produces dissimilar gametes. Male do not produce gametes. Male parent produces similar gametes. What do you think is the answer for this? Okay, I got my answer. B, ma'am. Pavan says B is the answer for this particular question. What about others? What about others? Come on, come on, come on. Male heterogamety, you have been said. Okay, motivation also says B and Nayanika also says it is B. So I have to log B, then answer is going to be. Why B? Listen, male heterogametic means males are going to produce two type of gametes. Here it is written female parent produces dissimilar gametes, wrong. Male parent produces dissimilar gametes, that means not so similar gametes, that is correct. Male do not produce gametes, arey baba, that is going to be wrong. Male parent produces similar gametes, that is also going to be wrong. So the only answer that we have is going to be what? B. Perfect. You guys gave me the correct answer. Now here I have the next question. Come on, try it up. XO type of sex determination and XY type of sex determination are the examples of. XO and XY type of sex determination are the examples of what? B again, ma'am. Okay. Nanika says B and Pavan says B. Motivational, come on. B. Can I log B? XY is seen in the case of Drosophila and humans. XO is seen in the case of grasshopper and things. Yes, they show male heterogamety. So B again is going to be the answer. Perfecto. Now do we have a question? One more question. I think last question from here. From this chromosomal complements given below, identify the one which shows female heterogamety. Motivation also gave me the answer B. Okay guys, last question. Last question. Come on. From the chromosomal complements given below, identify the one which shows female heterogamety. D, ma'am. Okay. Pavan said D. Nayanika also said D. Okay. Let's check whether that's correct or not. Motivational, I'm waiting for you. And others also. Macha, go ahead. Try. I'll not judge. Okay, motivation also said D, so that's why I'll lock up D. Exactly. ZZZW, this is seen in the case of birds. Here birds are producing, this females are going to produce two different type of gametes. So that means my children have understood each and everything which is up there. This is a similar question that came in second PUC. Okay, it's a 2-marker question that has come from this topic. What is haplodiploid sex determination mechanism? Mention an animal. Animal, you know, it's going to be honeybee. What do you mean by haplodiploid? Where basically, if the haploid ones, if it has one set of chromosome, it is going to be a male. If it is going to be diploid, 2 sets of chromosome, it is going to be a female. Then you explain about that. Okay, first of all, you say, in which the sex of an individual is determined by the number of chromosome set it inherits. In this mechanism, males develop from unfertilized egg and are haploid in nature. Females develop from fertilized egg and are going to be diploid in nature. That's all, and just give this particular diagram. That's more than enough. Okay. Pavan, so sorry, I'm not going to complete the whole chapter over here, but I have a good news for you. Okay, I really have a good news for you, and that is today. Okay, if you have all went through our channel, have you all went through our channel? If not, just give me a second. Okay, don't go anywhere, just give me a second, because I think it's going to be useful for all of you. So I have to go to YouTube. I hope I can open Diksha Karnataka over here. Okay, Diksha Karnataka is there. Okay, this is the class that is ongoing. Just give me a second. If you go to playlist, okay, if you go to playlist, you are going to see second PUC biology one shot. You see this? So apparently there are 3 one shots already, okay, on 3 different chapters: human reproduction, sexual reproduction, and basically reproductive health. Today you are going to get a one shot video on principles of inheritance and variation. This is going to be a very long lecture. It's like 4-hour plus video, but each and every KCET question and each and every second PUC question that have come from this particular topic has been covered. The same goes with all of them. Do not be so scared looking at the number, that is the hours. It is going to be in detail. So today at 7 PM you can expect that. Make sure you go and watch it. Will that work? Okay, and definitely I'm going to come in the next week, Wednesday, to give you the next lecture on the same chapter, but there's a one shot. Okay, hello Lata, I hope you're doing great. So as of now, actually I have one more live that I have to do here itself, okay, for some other purpose, which is why I have to go. I'll be listening to the class again then, ma'am. Okay, Pawan, thank you so much, and thank you, Motivational, and Lata, good to see you now, and Nayanika, as well as Motivational, as well as Akash, whoever was there throughout, thank you so much, and lots of love. Make sure you hit on the subscribe button, and the same thing you can ask about, or you can see for all the different subjects. Okay, sorry about, can you tell about pharmacy? Lata, what about pharmacy, dear? Tell me. And children, other children, you can go. Thank you so much. Lots of love. Please take care and have a very good day. What about pharmacy? If at all you ask me fast, probably I can answer you. You're most welcome, Pawan. Thank you so much for being a constant. Bye-bye. Okay. So Lata, I request you, okay, tell about pharmacy option entry. They are not giving many colleges, ma'am. So whatever colleges that is there, it's not, you're saying that not all colleges have come up. It might be because they have not given the seat matrix for those colleges. Because I believe whatever colleges are already there in the seat matrix, they have been put up. But if at all everyone is facing and lot of very important colleges are missing out there, either they would be doing something, they might be using something, because I saw these comments out there, but I could not find any solution for it. If KA extends the day or probably let us know about certain things, I will let you know as well. Okay? So, okay. So I think that works. Motivational, bye-bye, and Pavan, bye-bye. The farmer. I thought my mobile brightness was decreasing, but then I realized that you increased it. Oh my God. Okay. Thank you so much. I do not know how to respond to this, which is why I was just stuck. But you guys are the light in my life. So thank you. And bye. That's all. Bye-bye. Take care. See you really soon. Bye-bye.